Yeah, my name is Bo, and I'm a faculty member at Vector Institute, also a professor at computer science in ULT. I'm local.
Thanks, Bo, for having us. and yeah, we're very excited to I think start the discussion. There's really this the whole new angle. I don't know who is doing machine learning or AI to some extent. What about you? So I don't know how about you guys, but when I was talking to my family a year ago about these things, it's not that they're short stuff. But then this half a year ago, it was a big change, and interesting, and maybe that's also why Human Cell Atlas now has sort of. I think it might have something to do with Gary also sort of organizing. So we wanted to say, right, this is how classically cats versus dogs and supervised deep learning paradigm, and this is I think what we would need. Oh yeah, that's something. The audio doesn't seem to work. Can someone fix that? 1, 2, 3.
Fabian,
Yeah.
just see that the Zoom
I think
thing is turned on.
it is
Worked yesterday.
so we have now it should work. Zoom people, can you just type in yes, it works, or no, if not? Agreed, not so clear. Okay. Well, so you hear something, but that I'm not sure how to address. So we pick up microphones. They said this room is really quick. So.
You can mute them.
No, I think just I turned off the microphone. I didn't mute them.
Yes, and mute them, but turn on the mic.
I think I just. Okay, so we would just continue like that, and if someone can press buttons here to make this better, let us know. So what we wanted to say is that typical paradigm that we've been using is the unsupervised setting, right? To annotate cell types, find cluster, things like that.
Yeah, breaking up for the virtual audience.
Yeah, a lot of clicking noise. I mean, there was this.
Is it coming through the mic you're holding or the mic on the podium?
This mic.
Yeah, maybe just like turn off these and this. Is that better, online people? 1, 2, 3.
Yes.
Yes. Oh, okay. So we just dropped so this other mic. We just stand there then, right? All right. That's fine. Thanks. So yeah, this is sort of what we now did, just in the still mostly discriminative setting, like cluster things. But of course, the fancy and this has been a topic in machine learning for a long time to make generative models. I mean, even just simple mixture of Gaussian and something like that, super long time, but have been very effectively used. But in the unsupervised case, that's I think what people like. That's what's needed, and that would do at some point they give you maybe these nice things, so like how to put funny jackets onto our cells or something. So we would try to think. So this is what we want to discuss about. We want to discuss about this. I think that some people have been putting as sort of a next paradigm besides just the general deep learning embeddings that we have been doing so far. And this is an example we heard about yesterday, the Wang atlas, right? The integrated one where we have been, I think, taking something like 50 data sets, more than 2 million cells, superimposing them in parts using transfer learning to then add the data sets on top. And we have all kind of downstream use cases. So was it already foundation? So is these type of sort of encoding, decoding type of models or already foundation or foundational enough? Well, we don't think so, right? Because we do learn a joint latent space, but it does not automatically enable maybe even 0 or 1 short learning, depending on how much you want to fine tune to have then these downstream use cases. And this is, of course, what the more general community discussed. This is, I think, one of the big papers or preprints from Stanford colleagues, particularly this human-centered AI place they've been setting up. It's really a nice read. It's like a 100-page perspective type of thing with a bit of a biomed, but 1-1 as well. But this is the one figure that for me kind of stood out, and I think is a nice thing where you say, sure, machine learning we all know has sort of deep learning as a modern thing that I think most of us will be now using to optimize their models and sort of have them very flexible. And in some sense, you could say that you homogenized architectures in deep learning. Everyone's using one variant or another of a deep model, and that's why they have this emergence of and these features that in the last day were then, for example, used for logistic regression classifications, things like that, right? So you this feature learning from deep learning that has been a classical thing that been using, I know, featureize images of cats or whatever. And the point they made there, and I'm looking forward to discussion, Bo and everyone, whether these foundation models would then, homogenize kind of the models, that we don't now have a zoo of models. I mean, we literally call this model zoo, right? I mean, what you've been setting up near, but also you might see this like in the sequence-based learning community with the KP models and so on. These are literally called model zoos, and we have one with Star as well. You could say, if you now have one model where everything else sort of factors around that model, you just have to use this with one serial learning adaptation downstream. Maybe you have this homogenization, and that's why these functionalities emerge. So that was the idea, and you can take over and describe it.
Thank you. So yeah, this paper is really one of the first academic paper introducing, coining this term foundation model, and has been widely used in NLP computer vision since then. And what are some of the key features of foundation model? The paper has, I think, 200 pages, so too long, don't read. I summarize as A, B, C, D here for you. Key features of foundation model is following. A, algorithms. Most of foundation model based on transformer based. So they use attention mechanism. So it stemmed from the seminal paper from Google, attention is all you need, right? So depends on what encoder or decoder, you have different foundation model, but most of the foundation model people use NLP and computer vision are based on attention. Business B. Really the key feature when it comes to business or applications of foundation model is that one model that can enable multiple downstream tasks by transfer learning, or either zero shot or few shots. That's a general concept when it comes to applications for foundation model. And let's look at C, computing. And it comes no surprise that because of many foundation models are massive, so you really need to specialize hardware such as GPUs and DPUs. And I quote from Elliot, who is really the inventor of GPT models. He said on his Twitter saying, GPUs are the new Bitcoins. Just shows how important these hardwares are for building your own foundation models. And last but not least, data. In order to build a good foundation model, you definitely need massive amount of data for pre-training. You can have very little amount of data for fine-tuning, but when it comes to pre-training, most of the time we follow scaling laws. The more data you have, the more parameters you have. Generally speaking, if trained properly, the more capacity your foundation model will have. So data, everyone is saying data is the new oil, and this is definitely true for foundation models. And although foundation model has been extremely successful in NLP and computer vision, and if you go to any conference in AI, such as NLP and computer vision, almost 90% of the paper are about, foundation model or GPTs, these kind of things, or large language models. And however, in genomic, we really only start to see a few attempts in constructing foundation models in molecular biology. So here I use probably the most cited diagram in cellular biology, which is the central dogma. We'll find that from DNA, after transcription, you get RNA, and then translation, you get proteins. And for every modality, we have DNA, RNA, and protein here. Researchers from this field has started to construct some foundation model. For example, we know DNA birds, Informer, recently Hyena DNA, which greatly extends the context window from DNA sequence, are some early attempts in modeling DNA sequences. And for RNA, we have GeneFormer, my own SCGPT, and recently SC Foundation as well. For proteins, I was hesitating a little bit to put AlphaFold here, but still, many people, this is still arguable, but I think AlphaFold, at least it is a revolutionary deep learning model that use transformer-based model to modeling proteins. And also from Microsoft, we have ESM2, which has been widely used for a language model for proteins. So as I mentioned, for almost every modality in genomics, we start to see these early trends that people want to construct foundation model from massive amount of public data sets that is available to researchers. And once these models are constructed, people can take their base model and do fine tuning towards their own data set or task. It's still not widely accepted concept yet because everything is very early now. And there are lots of discussions about the pros and cons. And certainly this is a topic of this session as well. We hope everyone join our discussion about what do you think are the advantages, what are you seeing are the disadvantages. So here, Fabian and I summarize a few general ones. For example, we think one of the best advantages for foundation model is that it really enable efficient transfer learning. Fabian had talked about some of the work from his lab. We do transfer learning as well. And the second advantage is that it is more or less a generalist approach that it can use one model, the same model, deal with multiple tasks with zero shot or few shot. And the third advantage I think from my perspective is that it's really a good vehicle for data integration, right? We see multimodal GPTs in languages. GPT-4 you can just take a photo of your napkins and write some code on it.
Not yet, no.
Oh, good. I definitely want to see his.
It's only just started the second session.
Guys, could you just mute, please, if you're online? Thanks.
All right. Yeah. All right. Yeah. So, in my view, large language model based approach really is a good platform for data integration. And importantly, we put scaling law as both pro and cons because scaling law is basically more data with larger model shows better capacity. So this gives us a very good direction to explore. Let's put more resource into consortium like HCA to aggregate more data, and with more computing, we can build more powerful models. But certainly, it's also at the other hand, it's also a disadvantage because it concentrates more powers to those who have more resources. And how do we make sure all the large language model is accessible to everyone? How do we make sure individual academic labs can also utilize this foundation model, right? So we put scaling law as both pro and cons. And more disadvantage also, as I mentioned, in compute intensive pre-training. You need lots of GPUs and with lots of data with many days to wait. And also importantly, particularly for biology, it's a lot harder to interpret. For NLP and computer vision, at the end of the day, human is the ultimate referee. You can say this generation is great, this image looks beautiful, things like those. But for biology, many times ground truth is less clear to us. So how to interpret this bulky model is a lot harder. And also, we don't think large language model is causal model yet. So it's still doing a correlation and pattern matching. So how do we develop a causal method for this large language model is still unknown. For people online, could you unmute? Oh, sorry, could you mute yourself? Oh, this is
We don't have to.
we don't have the. Oh, we do have 71 online participants. That's great.
Not all of them are excited to listen.
Yeah. All right. So apparently this is a topic, a very early time, and there's lots of questions, lots of unknowns. So we list a few potential questions, but definitely this is a platform for everyone to engage the discussion. It's not just Fabian and I answer questions. Really, we want to hear your comments and questions, discussions.
So I think we organized, right? We have to put some question up. We can start with the first one, but if someone has some comments right away, I think that might be the best way to do it.
Yeah.
I'm most interested in number two. We're struggling as a lab. How do we represent and store our data? And we've gone down the graph route because I kind of makes intuitive sense to us here by eye, but I'm not clear on how to map a model to or a data model to a machine learning model. Do you have any advice or recommendations on graph versus other representation approaches?
In this particular point, we thought also how we then in the model implement that. Of course, there are models that eat up graphs, right? In some sense, with attention, people like to connect things again. But I mean, I mean, just like outline. I think for this sort of small scale integration project that we've been doing, there was not any particular graph storage something happening for spatial data. I think it could be good outcome if you want to do that. But I think I mean, you train on something like 30 minutes.
Right. We train on 33 million single cells. We simply store them as a matrix. People says you can make it even faster. You can store the tokenizers. So you save the tokenization step, but that's just.
So how did you make that decision to go with the matrix? Is it convenience at the end of the day?
Very good question. So it really depends on what data modality you train on and how much memory you have, how much storage you have.
We're pushing formats a bit, right? So if you saw in SCVerse, like this M data format has now also serialized version. So I think the bigger they get, we won't be able to have full memory anymore. So then you need to actually load parts of it in, and then I think maybe in your case it's still possible, right?
Yes,
Let's go closer to the microphone. Okay, so people can't hear people in the audience because they told us that everything has microphones in there.
So they can hear the questions from the audience, but they cannot hear your answers. So
Okay, so the microphones up there, not here. We stand here. Thanks, Alex. Yes.
I have a general question about the foundational model. So I guess in the classic foundation, or UNet or something, image processing, I guess that's one example I think of that is the idea is clear, it's classification. So here in biology, the foundational model would it have to be made with an eye on what the goal is? Like when you're making right now, the foundation model is the goal, the cell classification,
or can it be more general than that, trajectories, or would it be very specific and then,
Yes, so great question. So I think the key difference for foundation model in comparison to traditional CN like a UNet is really it's more or less a general purpose during pre-training. We want to learn the data distributions. Once we do that, then we can hook up the foundation model with different tests, such as segmentation, such as cell type assignment, things like those. The idea, as I mentioned, is one generalist approach. Once your model can understand, for example, what cells, or for in ALP, what is words, what are languages, then you can do all kinds of downstream tasks.
But that's why we kind of formulated also, I think you have a point here, right? What would be good use cases? So I mean, center prediction, obviously, I think one of the most common use cases, people just they want to use basically atlases to transfer labels and put on their own data sets. I think it would be fun to discuss also what people in the audience think, what more longer term want to have as use cases. I mean, obvious ones, right? Perturbation, maybe you also elaborate also.
So obvious for single cell is like a batch integration, multi-omic integration, cell type assignment, perturbation prediction. We also did a little bit gene record network, but we're very high level. So would love to hear your thoughts on what are the kind of specific use cases you are interested in and see how it works.
Just looking at second microphone as well. Yeah.
what's first thing?
Okay. I want to know that because it's also, for example, if we focus on cell type classification, it's also dependent on what data set you use. If we use adult tissue, it is very easy to classify each cell type. But in my case, I often use organoids. It's kind of developing brain. It's difficult to there's no clear boundary of cell type. So therefore, many cell type classification models developed by in the adult brain, adult tissue cannot be used for organoids. So therefore, I say it's difficult to
I mean, machinery, right? This is sometimes called style transfer, or sort of adaptation, right? Where your classical image-based things, you have the normal painting and then sort of make it paint as if it was somewhere in the over, right? And I think that you could potentially formulate also as a problem that you want to go maybe across subjects, you want to go across model systems, maybe even in vivo, in vitro, and these type of things. And I think early integration papers, even beyond foundation model, have been sort of trying SNGen and quite a few others. But I do think the point, and here would be interesting to see what you also think about,
figure one.
is, guys, please mute online, thanks. That would be to actually, by having seen not just human, but for example, mouse, right? And this is, I think, a reality. I mean, I think there can be some discussion how to sort of go just beyond only orthologs, right? But if you have seen enough of these situations, you would need to somehow to efficiently put that data together, sort of store them, and be able to find these connections.
Yeah, I agree. I think that's exactly the potential of foundation model. The idea is that you have. Sorry, for people online, could you mute yourself?
163吧，最少。100。
I'll try it.
没有127应该，我可以 check。
Yeah. Sorry for that. Yeah. So I think one of the potential for foundation model is that once you train your foundation model on very diverse data sets, it has a better generalization to something you never train on, right? That's kind of, people call it emergent phenomena, which I don't think we have found in biology yet, but that's kind of the potential for foundation model.
That's a good point to add to our use cases, right? So let's say organoid, or like maybe even maybe cross species. Yeah. I think it was Christoph, then Malte, and then.
I think these foundational models also force us to really think very carefully about what we measure and what we want to do with it. And it's clear that we have, opposed to the large language models, we have kind of two different domains. Large language models is learned on text, and we want to write text. Great. We learn for the most part on homeostatic states, cell states in the normal human being, evolutionarily optimized states. And what we want to predict for a significant part is perturbations, where things is moved outside of the homeostatic state. So there's a fundamental disagreement what we mostly train on and what we mostly want to do with it. It's a bit similar, you mentioned AlphaFold earlier, and AlphaFold is also very good at predicting the homeostatic ground state, but it's quite bad when you try to use it as a protein design tool. And people have really kind of then started engineering mutations into AlphaFold, and that doesn't quite work. You cannot predict how kind of a single point mutation in AlphaFold affects the structure, because that's kind of what AlphaFold has done very well, is taking into this evolutionary dimension into account, which we have not yet. So I think we need to, this would be kind of my two orthogonal themes. We need to be very deliberate about homeostatic versus perturbed state in training and in application. And we need to be very deliberate about the fact that we are evolutionary products and modeling, incorporating evolution.
The important point, I think, let's say the more perturbed data set that could be like literally perturbed or something in an in vitro situation, or maybe also, in particular diseases with trajectory variable, I would expect models then to see more of that sort of complex landscape of states and then be able to go out distribution. This is essentially all out distribution. That's like the hard problem, right? And, I mean, if there's paper, what would it be, these sort of regulatory-based ones, such as what's called from Sam Morris's lab, Cell Oracle, right, for example, right? And these, those coordinates that underlying regular structures really robust, so they could potentially have more access there. I think the point that you're making, that would be good to add to our list of use cases. Yeah, train on transitions.
the audience, audience as well. And after you
Hi, Jasmine. So this is kind of on Christophe's point, is one, if we really want to apply this generalist kind of idea or tool, even what we're feeding it from a HCA perspective, right? That idea that we're creating a reference map that you can now plow onto disease. We haven't incorporated, they're all static states. They're not cell agnostic. And then once we load on spatial, right? The very idea of spatial being fed into it, one becomes a huge infrastructure problem. And two becomes like, how do you incorporate all the reference, all the training, all the pre-training we're doing on these foundational models to things that don't exist, right? To things that are evolutionary different because they're in a tissue and they've incorporated some static, but some evolution. And how do we feed that into the system as a collective community, right? Because we're all keeping our own versions of a gene count matrix, right? We're all doing that because we don't have enough space. So how do we collectively think about that in terms of a true foundational model that we can't really do in the same way we're doing a reference map for HCA?
This connection, I think you raise a really important point, right? Like in HCA, in some sense, we want to put together all the data in the world for at least describing maybe stack, but there's going to be developmental atlas. There's discussion about perturbation atlas, and some of the whom, they're pushing for these directions. So I guess if we keep the data, one discussion point, I think you actually already wrote it, right? This is our point 8. How can, HCA maybe as a community also train this, right? So that's not just like in particular labs, that this is also being shared and doesn't become like a company then owning that, and we just need to sort of go where they're more.
Because we need the data, right? The only way any of this is going to work is if large efforts like this are done. Where we've annotated the meta. Spatial is like a ridiculous problem.
Yeah. So spatial omics definitely a very interesting topic. Lots of people reach out to us saying, can you extend to SGBD to spatial? It's actually not that straightforward. We were thinking about it, but first of all, we need to spend lots of time curating the data set and think of, and different technology have very different characteristics. So that's a challenge. And some of the HNE images, they are very different resolutions. So that pose additional challenges. But I think really the first challenge we face is data set. And then how we were discussing with HCA and Fabian, how HCA can guide us about curation of data sets. They did a great job on single cell RNA, but that's very limited scope. Can we expand to further modalities?
Maybe we take an online question first to sort of get a bit of a beef in the room. So Dianma, and I think that Dan Burkett is also enforcing it, asking a bit, why do we even need foundation models in this context? So there's this argument, would it be a sort of fit for purpose model that says specific still do the job better, right? Correct.
Yeah. I don't think we need a foundation model for everything, though. I don't think that's true, though. Even for GPT-4, it's although it's probably the most powerful AI model so far, you don't need GPT-4 for everything, right?
So would you really say that nowadays there will be any new model coming up that deals with language as input that does not use an big LLM?
It's definitely a lot harder to publish. You don't need large language models for simple things like
true, if you want to run the
exactly, yeah. So, but if you really want a model with large capacity in languages, the general practice is you take a pre-trained model, either Llama or other GPT variants, and you do fine tuning or zero shot or do in context learning. That's a general practice. I'm not saying we should do that for biology, and we already have lots of fantastic methods from this community that can just stand alone and work on your data set. I think the purpose of foundation model is that, as I mentioned, when you have very little data set and some of the traditional deep learning, for example, STVI or CN, they cannot deal with this small sample size. Actually, fine-tune foundation model is actually probably a better choice, right? Or if you want to integrate multiple modalities, I think in comparison to CNN, attention transformer-based architect has better flexibility in integrating different modalities. You probably heard prompt engineering. So we could do similar things in genomics too, right? Let's say, can we infer certain taxic data using RNA-seq as prompt, right? Because you can, from architect point of view, you can use cross attention to bridge that two modalities. So from architect perspective, it's much easier than CNN from latent space. So that's kind of the advantage I was thinking, but definitely when you have enough data, you have enough prior, I would choose whatever that is available to you, right?
There's Barumia asking online as well that there's limitations to current reference genome, cell type detection. We might not find rare cell types, and it goes a bit in the direction I think what Christopher was saying before. If you don't see particular transitional state, you maybe cannot train them there. And they ask about the efficiency of transfer learning. I would argue to that point that I think there's loads of papers already in the normal set of deep learning setup that has shown that I think transfer learning will be the way how we want to use atlas. Because I mean, like, what do we want to do next? We just don't want to build an atlas because we want to say, hey, we're done. This is the atlas. But it's like in human genome project, once we have assembled it, we want to use it as a reference and map our stuff on top. So it's like, for me, it's the basic use of transfer learning. I guess there's limitations, of course, but in my opinion, the need then to address this would be that's an uncertainty model. And this is maybe something that I think also current models struggle with in language side of division. And that's why so the reinforcement learning a human loop in that. I think we also call it some ideas there to take as
So I had a point regarding some of these very interesting discussions, particularly with out-of-distribution generalization and kind of new discoveries in biology that we're looking for. I think one of the challenges as a community we need to keep in mind is how to really assess these foundation models for that kind of generalizability without this kind of implicit data leakage because of, let's say, amino acid sequence similarities, DNA sequence similarities that you may have been very careful about setting our test set, but it is leaking data to kind of indirect data. That would be one of the most difficult challenges that we think about.
Yeah, so that's excellent point. I can emphasize how important evaluation is for these foundation models, and data leakage is definitely one of the concerns. That's why, at least for our SCGBT, we keep a good documentation about which data, which cells we train this model on. So at least we know we can go reference back saying these are the cells we train on. So if we have part of the cells, that's probably data leakage, right? But keep a good documentation, good records about what data your model is trained on. I think that's first step. Second of all, we can draw inspiration from NLP side. They have various leaderboards that you have different tasks and different metrics to evaluate your large language models. It's not very clear to me how we can do that in cellular biology or in genomics. We have some existing benchmarks, but I think that's also part of the discussion is how do we find novel use case to verify how generalizable your large model is, for genomics.
We had discussion about communities yesterday. I think the organizer and I think one community aspect that could be used for this. I mean, there's these obvious things such as Kaggle, right? Or there's this open problems.bio and related things. You have to aggregate metrics. Typically, if it's public data sets, they would have been leaking already in such a model. So it needs to be done for actual competition, some newly generated ones. I use particular lab effects, so batch variation to when there was I think Malte had a question over there. Oh, you go ahead first.
Yeah, just a few quick points. I mean, we've been using machine learning methods and the thing is that the community generating additional supervised learning, you're trying to make accurate predictions. You're not predicting them to apply. And if you have a regulatory model predicting expression, generally it's not predicting expression principles, right? It's to understand gene regulation, right?
Correct.
It may be perturbations, predicting concerns, but that's not reasonable. We're not just trying to predict acceptability, we're trying to right? And similarly to new large language models, we don't need to generate facial pictures of tumors, we have them. They're from actual patients. We're trying to understand what's wrong. So I think, you know, the goals are different. And
Friendship privacy, I'm sure there's somebody who actually want to do that.
Generate a tumor.
Something that they can share the machine with people without doing HCA or whatever.
Yeah, and I would say there's a bit of a disconnect. Like I'm also very excited about the possibility of large scale machine learning, but I would say most practitioners, people who are actually trying to solve scientific problems, are using machine traditional statistical methods to analyze all these. But it seems there are more papers than users. So that's telling us something that we're not reaching our audience, not as much as and the final point is, you know, what biologists do, good biologists, right? They actually reason about the cell, right? So they have access to all this understanding, and they're trying to understand what's going on, or how our cells are interacting with each other, or how the immune system work, or how is the cancer cell surviving and adapting. And we're not really accessing all of that. So if all we accomplish is label transfer, that's good, right? But we're not in biology.
Yeah, that's excellent. But I think most of people here probably computation biologists, so that's a constant struggle for sure. What I do most of the time is collaboration with lots of biologists and understand what's their burning questions and design models specifically for their questions. And I think that's and also probably HCA can also play a role in bridging the gap between computational folks and biology. And yeah, I think that needs a community work. But I do see an uptake of computational approach in biologists, right? You probably heard yesterday's keynote by John Dick. I'm a close collaborator with him. They use lots of machine learning. Even
you do all the GC first? He was showing.
That's part of the work from our lab too. You know, so I think, as I mentioned, the collaboration is extremely important.
So I think you described like a general trend, like in machine learning. Obviously, the content explodes. So many people publish it. Many of these things are not being used in past because just the code has been engineered. I do think though, in our field, like with the explosion of so many labs doing single cell analysis, that I think our tools are really needed. But I think the point making them actually attach to problems. I think most of the discussion was what type of new problems could we actually chuck on top of these and see if they really factor our foundation model with specialized models. Those questions might.
I noticed that a couple of times where it's been mentioned that data, right? We need more data. It's context, Chris mentioned, I think mentioned also, you know, to be able to train, like, to learn something out of the distribution for these models, we need, you know, data to cover sufficient numbers of kind of distribution in the first place. It strikes me also that we haven't really talked about data quality. There are several kind of papers in general in machine learning, you know, early, looking at important data points early, or reduce training time. When we're doing an integration task in the integration team, we start with a lot of data sets, and then those get smaller and smaller because some of them just don't integrate well because of quality. If we need hundreds of millions of cells, there is a tension between capturing as many different scenarios as possible and filtering that out better quality data. I wonder how you think about the data quality, because I don't see an LDM because like every third word is misspelled and you have the next instinct. So that's what we
If I take a first step, I totally agree with you. Data quality is actually very important. So if you look at SCGPT, first we manually curated a large data set that has, I think it's 10 million cells. And then on the same week, we put the preprint HCA release cell by gene curated data set, which has 33 million. And then we, without hesitation, we switched to HCA curation because we believe their data are higher quality. And we do see difference in terms of training, particularly as you mentioned, early training phase, and we can compare the curves. We will find that train on 33 million cells with from cell by gene will give us much better capacity. And so that kind of reflection of one is number of the diversity of the cell types. Second one is the quality of the curation, right? And again, I want to kind of involve HCA. That's definitely needs a community work to make sure a good data curation, good data integration pipeline, or at least collection or quality control are in place, and people can train their model with more confidence.
So short further comment on that, because I do think in language models, right, it was, I think, clever not just to train on all of the internet, but then, like what's the Reddit equivalent to us? I mean, should we just look at Alan Brain because they to make everything super harmonized and very nice quality? I guess it might be better for brain data set. But then for these obscure states and transitions, leaving models robust enough to think. I think this is basically also what needs further iteration. We should take an online question. So the question is how to go make use of different data modalities. We had this point, I think, also right about cross-organ, like multimodal and so on. How to use these in these foundation models. Language model like GPT is straightforward. There's no data modalities. How to deal with this? You want to get a step first?
Yeah, sure. So I think I briefly touched upon this one before. Is I do think large language model is really great vehicle for data integration using the cross-attention mechanism. So we will actually have some ongoing approach to integrate ATAC-seq with RNA-seq, and the cross-attention mechanism. That's the architect we chose. That's also how you probably heard stable diffusion, mid-journey. That's how they do it. They do cross-attention with text embedding with, and also to generate images, to generate languages. And we certainly need peer data, which are relatively lacking in this field.
Yeah, but not on that scale, right?
Not that scale, yeah, exactly. And there's also some techniques to use unpaired data to further training. I think there's ways to pursue that direction, and that's kind of one of the reasons I'm bullish on large language model, because I think it's really from a computational side, it's a good vehicle to incorporate many other modalities.
I mean, stable diffusion, the guy who develops actually colleague in Munich, and they've been in fact not having sort of super large resources for paired data, right? So it was like a few, and then they used to train language models. There was a question.
So I guess, thinking about data that we need to be generated in order to help with these foundational models is tissue samples that are not at end stage of disease. So much of our disease states are coming from cancer when it's stage 3, stage 4, in transplant when they've already experienced rejection, in autoimmunity when autoimmunity is onset. So, if we're going to build a foundational model to understand how these diseases are progressing, getting activated, we're really missing those earlier time points when it's being initiated, right? So I guess a question for you both is, if you have, homeostatic and disease, how well are these models at dealing with all the steps in between A and D, right? And that's a big challenge. And then, I'll leave it there for now, actually.
Kind of harks back to the old days of pseudotemporal ordering, right? Where had this one, maybe a few styles which could help transition. Could use that. People nowadays use a lot of time series information and can integrate these. That might be built with foundation, also more classically around tumor transport for some reason. Yeah, if you get more of these transitional states in, from my point of view, I'm quite hopeful for the development of maybe even doing this for specific tissues such as brainers first, because there you do see transitions and maybe see the hark back to some of these mathematical states that people mention.
Yeah, I agree with Fabian. I think we need to put more data out there, particularly for developmental and time series data, which I think the community is doing similar things. So I'm hopeful that once we have data available, that's like not just foundational. I think that's true for every computational approaches. Once we have the the right data available, I think there will be no shortage of methodology.
I don't think we need to repeat the questions because sometimes people don't hear the questions I heard. There's a next question. Sorry, hold on, then,
so my question is related to which other ethical concerns are involved regarding foundation models, and how can we add regulatory elements that actually like restrict that in some way. So we don't want the foundation model to be aggressive in a way as well, for just like, as an example, or suggest to do things that are kind of not correct. If you could comment on that, thank you.
There's 2 points to it, right? So maybe I take the ethics and I could take the hallucination one, because what you're saying, it could be like going continual through into bad stuff. I think for the regulation one, I mean, something like GDPR, I hope I'm saying this correct, for just like new AI laws coming up, which could be potentially quite devastating, that users should be allowed to retract their data from a trained model. So you spend a lot of effort into training a language model, or whatever model. The language model is even worse, but in our case, it's starting to get big as well. And suddenly you need to remove one thing that's going to be hard, right? And it's going to be destroying the industry. So these things, I think we're still in a bit safer side, that we work on the count metrics, where people are maybe not yet wanting to argue exactly about that, but as soon as we would be adding genetics, and I think that's the next thing. I mean, Christine, you mentioned this already, right? You want to add regulatory part, and as soon as there's going to be steps in these things, it's going to be very difficult to deal with this, and it might be then all with managed access and so. But that is something to keep on the radar very early on, and then have to sort of set up an application. Now you asked also about the new things about the hallucinations. Right?
Yeah, so correct. Just also comment on the previous one as a safety issue. I think it's important to make sure the model is transparent and open access to everyone. I think that's first step. And make sure lots of researchers in still trying to understand what's happening within the large language model. So I don't want to really regulate the development of AI. I think it's very helpful, very beneficial to the whole society, genomics as well. I would rather regulate.
Are you from Toronto, Hinton? And you're saying, I want to take that up with that guy.
I think we are still early phase, but at least for genomics. So yeah. So, but definitely, I think it's good to have this responsible AI, this mindset from the get-go when you develop your model is very important. Regarding the hallucination, I think it's definitely an issue. It's not even solved in languages. It's really the holy grail, to be honest. People, to be fair, hallucination, the issue of hallucination exists from the get-go of NLP. People didn't care about it because there are so many unsolved problems. And now, GPT-4 almost solved most of the language model issues. Now people trying to tackle the most challenging one, which is hallucination. And so it's definitely an unsolved problem for NLP yet. It's less clear to me how genomic model such as like a DMBird or informal or SEGPT hallucinate in this context, because we are not so far focusing too much on generation yet. So it's less vulnerable to hallucination in that sense. But also, I think Aviv yesterday has a point that I would be very interested to see how this model hallucinate, because there's a spectrum that for the extreme of this side is fact-based. Everything is controlled by very specific domains such as search, retrieval. You have a database, you retrieve. You don't jump out of the database you have, right? So everything is fact-based. Another extreme is everything is generated. You have extremely high innovation. You generate things even if you don't know what it is. That's hallucination. People want to solve that. I think NLP's the current GPT model is more or less in the between, and you don't want to remove hallucination 100% because you want some generative power, right? That's exactly why everyone loves GPT-4, is because it can innovate, right? You don't want to lose that ability, and human hallucinate too. That's what makes us innovative.
I mean, that's pretty much, I'm fascinated by biological partners. Whenever they come up with a new experiment, they sort of by serendipity and this complex graph in their brains that at some point, the brain tells us how this works, come up with the next experiment. So I think it could turn into a feature, right? You could like say, hey, hallucinations, experimental design. No.
How do you know when it's a good or a bad hallucination?
That was the thing we had on uncertainty. So basically how to really make these models uncertainty aware. That would be really important, right? And I mean, maybe you also want to comment on that, but no, this first embedding models, we just had like this normal autograph, and you just sort of make things variational. And there's this whole community of Bayesian deep learning where people like go full distribution. That would tell you the full posterior. So then based on that existing data, I'm sure that this is a good thing. This one is maybe not so much. This is of course much harder for.
This is very hard for large language models.
One question from Rashad and then maybe a couple questions.
You mentioned a lot about data size, data quality. But in NLP, which we have simple grammar rules, they train on 2 to 5 trillion tokens, and it still cannot do a simple logical multiplication. If you do ChatGPT a simple multiplication, it doesn't give you a good result. So I assume a foundation model should understand the underlying biology or biochemistry of the developmental process. So I was wondering how, which we don't have that trillions amount of data, how can we, with this type of training, with just simple math, we expect to discover this physical laws?
But you actually are doing masking. In this case, that's auto-regressive.
Yeah, auto-regressive. But there's also I see a very good question. To be honest, I don't think the current all the language model we mentioned here can solve the problem we mentioned. It's really just to understand the data distribution by far. And I think the key missing piece. If you look at it, I want to again draw the parallel to GPT-4. So GPT has been, large language model has been in development for many years actually. And why all of a sudden in end of last year, ChatGPT came out, it's so much better than everyone else. It's because the introduction of human feedback, reinforced learning on top of the pre-training. So far, most of the model we are talking about only do the first part, which is pre-training, which is data-driven pre-training. We want to predict what's next, either next word or next genes, next DNA base, right? So we don't have instruction fine-tuning yet. We don't have human feedback loop in the loop. So I think a good direction, I think at least my lab is thinking about is how can we involve priors from existing literatures? Because that's text. And there's lots of knowledge graph out there we can use as a prior or either regularization or prior or retrieval with verification, since I know.
So that's actually, I wanted to say, for example, I think point 3. So
Use
why?
microphone.
So like we have this literature of, as you mentioned, physical laws, all of this. So why don't we add them to this, so we make this model controllable? Like for example, if your different paths go from A to B, we choose the path that actually is based on a physical law. So the loss functions, you're using autoregressive or math inspectors. But you actually like make it more complicated loss function, you're now able to control a lot of things instead of even the reinforcement.
The question is, right, what are our physical laws? You know, I mean, you can tell it, go to the calculator and suddenly you have the calculation, right? I guess for us, maybe the dogma part, that could be something like, you know, integrate ATAC and RNA or like, you know, unsplice, splice, some of these things. This is what people would believe that could be true. Apart from that, I mean, you said knowledge graphs. You don't want to like hard code it. You want to
correct. Yeah, we want, I think the way to incorporate knowledge graph is more or less like a retrieve based. You know, for certain gene tokens, we retrieve the nearby gene or genes that interact with, and then find shorter paths in the knowledge graph as a verification to one, reduce the hallucination. Second of all, is to improve the, as you mentioned, to more abiding to physical laws. But technically speaking, I think it will definitely complicate the training for sure.
Yeah, for sure. Yeah.
You know, so but definitely interesting direction.
Should take an online question. Hannah Ali is asking about when we say massive data, how large should that we, for things applications, we already have enough data to train a foundation? Kind of goes to our first question, do we already have foundation models, or are they? So are they outperforming linear models for cell type prediction?
I can answer that. Generally speaking, I don't think we have a good way to do power calculation for large language model. Generally speaking, it seems true that the more data you have,
well, we don't bring cash or credit cards and that.
Somebody's already to pay for his foundation. Calling out different discounts. In those,
All right. Yeah, I think Zoom should have GPT-4 to anyhow. Back to the data size. Generally speaking, it's what we see so far is the more data you have, the more parameters you have. Generally speaking, the more capacity, more higher accuracy for downstream tasks. But the question is, when is enough? I actually don't know because when we plot the scaling law, it's still uptrend. So we see some diminished gain after 3 million cells for single cell for SCGPT, but the trend is very clear, And also maybe that's just for that data set. So who knows how new ability they can unlock. So I actually don't have a good idea about when is enough for data trend.
I mean, we have to be honest, right? Even for the simple models, like, how many of you has actually done a power calculation and who hasn't just looked, what's my next year's budget? And it's just, I mean, like what's the number of patients I can get or things like that, right? I think we should be at some point doing this. I think particularly if you talk about, we call it human in the loop, I think Aviv is calling it lab in the loop, and this is of course an old idea of experimental design. I think maybe these things should become possible. But even I think for the simple models, we want to take one more online question and then we can go to the room. There's Mark Zaidi asking what our thoughts are on how to approach standardization of image formats, okay? Because you asked about the data part of it. I think in the end, so much is about this data. Means that not having the ability for foundation models to read image of training, but also any other relevant metadata. And I think this is a beauty, right? We have in our fields, if it's just about genomics, we kind of mostly have the standards. There might be a bit of discussion about reference genome, but that's about it. But the more we go into these other formats, it gets worse. And I think if you then go to medical imaging or so, there's like all these DICOM sub things, it's going to be a mess. I think already now in our field, maybe, and data, people can hide a lot of things. That's maybe not so directly clear, that's sort of standardized, so that's why I think Sabatin is useful also CAP. But for image formats, I mean, I have brief comment on that. I do think we should not let loose of it. Whatever we publish in HCA should adhere to some standards. So in fact, we had discussion yesterday in our working group whether to potentially have some recommendations there. I do think that could potentially play into that discussion we had yesterday, just take that up again. But yeah, it's necessary. Are there any thoughts, I mean, on image formats here also in the room or more general sort of need? Because I mean, disassociated what's can we most get, but there's like there's not a clear winning technology on the imaging side. So we hear back from AWG working with an HCA. I hope that works, Mark. There's another question.
Sure. All right. It's a great discussion. I'm wondering about a legal aspect / copyright aspect of it. I mean, for text and image foundational models, there's a lot of lawsuits going on, a lot of people that are being, artists that are being copied. And in our case, we are not dealing with cheap data. Like, it's not images or text that anyone can generate for free. It's just like huge data sets that take a lot of money, a lot of time, and people that are basically paid in recognition. So how do we ensure that these foundational models will give back to the scientists what they need and to the funders and so on?
100 cells in same cell state. Now you have a better grip on comparing batch and biology first, just aggregation itself.
Just listening to both, maybe.
Yeah, great question. So first of all, the copyright, even different countries have different regulations. I know probably Europe has more stringent regulation. Japan recently have a very landmark ruling saying AI does not violate copyright. But for genomic, I think we're still far away from that discussion yet. And once
citation is the point, right? That's
yeah.
sort of our copyright.
Citation is a good one, and also open access, right? So far, we make all the models, different models, accessible to everyone, and we also code tutorials, things like those. Hopefully, for every foundation model we train, we can keep that, so that this is foundation model for everyone, right? Everyone who wants to use that should download, but also keep a good catalog of what data they are trained on, what hardware, what use case you think of maybe suitable, things like those, just make it more transparent.
So this is a general problem basically of all kind of aggregation approaches, right? For these first integration papers, there was already 50 data sets being used in the long atlas to put together. And of course, you won't be citing those 50 papers anymore when you do your cell type transfer. There might be one particular big one, and it's kind of then not happening great. So I think one needs to be transparent about these things. On the other hand, of course, in most cases, it's also taxpayers' money. And so I do think, and it's not in all countries the case that people don't publish. So I think also these models at some point could maybe then go to another tool and then they're going to get cited anymore. It's just this back and forth. And I don't know if in particular more in medical literature, there has been this discussion of data parasites and so on. It sort of precedes this type of discussion. But I do think I don't think that will be this long-term problem this setting, because HCA openly contributes. Again, the idea is people want to put the data sets out there for them to be used. And yeah, I guess you need to sort of Sabagene explicitly tells you this is coming there. If at some point there's going to be attribution techniques coming up, and you say that this sort of one particular cell has been taken from that particular data set, then that could be actually then leading to a discussion in that.
So you mentioned for the LLMs, much of the power is in the fine-tuning, human-level fine-tuning. And I think that's very useful for us to consider that from the beginning, especially that at the moment, much of what we know about biology is in kind of the brains of very well-trained biologists for each specific area. So whenever we engage biologists for cell annotation sessions, for anything, we should keep very good records of those interactions, because those records could be a very valuable resource going forward. Because kind of anything where a biologist, like a domain expert, is at work interacting with the data. Maybe for some of these cell annotation jamborees, that's relatively straightforward. You want to kind of, in the extreme case, you want to do kind of eye following. That's probably overkill.
Because at the moment, what do we record? We just record what cell type they're giving you. That's maybe not enough. So you're saying that's much more, right?
You want to you need to find kind of an ethical and doable way that people are actually willing to consent to. But things like, ideally, you want to see who they Google on the site, how long they
store something on the laptop, right?
And how long it takes to assign a label and all of these data of the process, because that's the only thing that will not go away and will not scale. Is kind of human time at the machine. Everything else will eventually be solved. So whenever we have a chance to record and some of these domain experts, like biological neural networks of real biologists trained 30, 40 years, we need to capture that while we can.
Yeah, this is excellent. I think we just lacking a platform to keep track of this thing. I heard an interesting study from James Zhou, who is a professor in Stanford. What their group did is very interesting. For some reason, pathologists like to discuss pathology images on Twitter. I don't know why, but somehow they like that. And what James Zhou did is they download all the discussions, interactions between pathologists and the corresponding images. They construct a language model for pathology images. So similarly, we should do that for single cells. Maybe we should do that. We have enough Twitter engagement. but somehow, I think that's definitely a good source of training data for sure. That's an excellent point.
I mean, to be honest, also what you just said before, right? I mean, also the papers that biologists in the end write, you know, this figure 7 or this graphical abstract or things like that could potentially be there. But I think this at Jamboree in particular, because that's something that we can actually influence. It's coming up now. Maybe Malte, you have a comment on that because you're organizing that, right?
It's a really good question. And there are still efforts to include this. You know, Chloe is their CAP is literally trying to record metadata of the rationale for each and so this is, you know, they deliberately make it very general what metadata is. So it can be many different things. Also for the HCA, we started not only recording, you know, what labels people gave, but any comments they had on that particular cell, plus markers they used for this. And so I think there is efforts to start building a situation, to start capturing the situation. And of course, you know, there's many things you can still do, but I think the CAP is really designed.
And the machines will be so much.
The machines will be so much better. So if you record now all interactions at this Jamboree and you cannot do anything with it for the next 2-3 years, eventually the audio processing and interpretation will be so good. Like we should record much more kind of verbatim biological interactions. Just put it on file for in a couple of years when this will be really, really valuable because the methods have advanced sufficiently so that all these raw data, let's record kind of biological human interactions as much as possible because this will be extremely valuable in the future.
Ellen, is this recorded here?
It is recorded and we have an archivist who will be transcribing it.
And I think this click part, you know, in CAP, for example, how long people actually sort of need to think before they assign. It's like a very low-hanging measure to do, and that's like directly usable now. But the other stuff don't work.
So let's do one more question from the audience. Then please take a look if there's anything on Zoom, and we are scheduled to be done in about 8 minutes.
I have a question about this discussion point 4. What is the best scale or best way of the cross-organism, cross-species analysis? I think most of community will obtain that some orthologous gene list and pick up, combine that the count matrix table from human and the mouse and for some normalization. But I also think this is really best way or not, because most of goal of cross-species analysis is to identify species-specific feature. But this orthologous-based approach removes a lot of human and mouse specific species-specific gene. Therefore, yeah, common, you know, we can identify common things, but it is much hard to identify some human-specific features. So how I wonder how community think about the best scale or best way of cross-species analysis.
I was just saying this briefly before. I think this is an important point, right? At the moment, orthodox, very simple, but it's not capillary tolerability, and this whole dream of Evo-Devo has been a very big thing in knowledge for a long time.
I think
right?
several thousand genes will be removed.
I think there's more data coming. You know, we just don't want to like put them completely separate spaces. There's nice poster presented here from Leskovich lab on Saturn that uses protein as a language to connect this. But there's I think different ideas. I have one industry question, but I think Roma, you asked quite a bit before. So let's just take this. Could we take this briefly, and then we take one online question? Then I think we're done in 5 minutes. Is that okay? Okay. So is that another take the online? Yeah.
Thank you. I wanted to ask. I think so. We're discussing about coding discussions about biology, like recording in the room. I guess we're already taking pictures, so we don't have the voices here. What we haven't maybe quite discussed. Video. What we haven't quite discussed is how to think about the context in which we're doing experiments, and maybe like there is some software used in like labs for recording context and like what cell line we're using for generating those experiments, how long we're waiting between, if we're doing perturbations, like how long we're waiting.
The protocol documentation.
Exactly. Yeah. And the key thing would be. Or even like when we're analyzing this data, sometimes we're having cell level annotation. It can be just like descriptor of cell types, for example. And all of this is also like text in covariates. So how do we think about this and adding this into those language models? And I think to me, this is important because we want to think about commandability. So the way it's usually done is question answering, and we've been talking about that a bit. We don't know how to do it quite yet, but in that specific context, if we start having like descriptors of cells and conditions for experimentations, and we start understanding like what is a protocol that's been used for aligning the counts and aligning all of those reads, what's been used for processing all of those cells.
Okay, so even the computation protocol.
This could be everything, yeah. And would that be a way? Because once we start having like tens and tens of data sets that have been processed with SmartSeq2 and tens and tens or hundreds of data sets processed with 10x, can we start asking questions like, oh, what if that data set was generated with 10x instead of SmartSeq2? That's also when you get the hallucination, but that's actually like when you can get to generate things. What if that cell type was also perturbed with activation of like that virus, if you have like that kind of experiments? And it goes beyond this, like just encoding cell state, but also thinking about the features. And it creates huge problem when you start. So the way that those models, I think, have had an impact, I mean, they're starting to get a lot of attention, is because we've been able to get these really nicely clean data sets, cell by genes. But when you start thinking about the way that we do experiments and that it's not clear what is the best assay for special transcriptomics, that's going to change over the next like 5 years like crazy. We have no idea what the assay is going to be in 5 years. So is there a way that those models can also accumulate data from maybe slightly changing protocols across modalities? So that we are able to actually integrate data along the way when technology changes as well, instead of just focusing on sequence RNA with cell by genes.
I think that's a excellent question and suggestions too. I think so. Fabian mentioned like have a good records of catalog of protocols, right? Even like a text-based. I think that works too. Or covariant modeling kind of approach for that too. Another idea I want to bring, it may be. So you probably heard that GPT-4 recently introduced code interpreters. That means GPT-4 now can generate code and run the code for you. So it's probably too early to discuss, but I think one potential is that we can combine language models that works on omics with language models such as GPT-4 into the day-to-day bioinformatic pipeline for biologists, so that the language model can learn from the protocols which are text or even audios, of the things we are talking about. And the omics learns biology, and the languages keep records of these biologists, documentations which are text or audio, things like those. And then, but certainly we need to figure out how to combine these different language models. But I think it's an interesting idea.
We've been discussing this in the lab as well. I think also in the general community. Remember when we had discussions, how do we use HCA? And it was like, where's my gene? Where's my cell? Where's my gene program? And maybe we can formulate better things in natural language at some point than put that on. I think the covariate space, I mean, you have the Mr. VI, like some of these things sort of make this more rich. I think that's actually a nice general setting. I think it applies as well. If we could take like one last question and we wrap up from the chat, is that okay? Sure. But you had something to say?
Yeah, I did just maybe.
Yeah.
So okay, I don't want to. I know that we need to wrap up, so I don't want to open. So first of all, thanks for the session. That was very informative and fantastic discussions. And I want to echo Christina's point here about how we make these models useful in the actual research that we do. And I think one aspect to that, and I'm really curious to hear your opinion, is about experimental design. And how we can make recommendations right from the next experiment, and this is really connecting with the hallucination of generated part of the model. So that's one of the question.
Yes. As we discussed before, I think that's the thing to do. I guess if you have a big company, you can potentially even wrap this around and. Yes. Hi, 1, 2, 3.
Yeah.
We're about to wrap up. So the last part would be if we could get people to So if you plan your next experiment, maybe try to query, let it be an LLM-based or transformer-based model, even something simpler with that, and then see how these loops could be done. But I do think there's actually even still quite a few maths to be done. So like, you want to do multiple experimental design conditions at the same time, right? I mean, that's like, if people just want to minimize overall uncertainty, or is there something more specific, maybe with prior knowledge? So I think it's going to be for sure papers coming up more in this direction. I remember like 5 years ago in the white paper, we already had experimental design in, but I think that's been like literally nothing. It's like SC power, but it's maybe it's one more like Raoul's how many cells versus all. So I think it's good to pick it up. And maybe the last point with this industry. Yeah. So Jean-Baptiste, you asked that. I mean, Bo, I think you also see, right, that I just read a state of AI last year. It was say literally all the large models that are run in the industry, not in academia. Right? So I think that's a point. Maybe we also want to give a comment on.
Yeah, I would love to have more industry and academic collaboration because we can share data, share resource. But hopefully the collaboration is built on the principle of transparency, open access, because you don't want the concentration of powers to certain private companies, and then academics don't have access to those resources, right? So definitely, I think it's definitely a good idea to collaborate with industry, to have collaboration between industry and academic, but we should set some kind of principles for these collaborations so that everyone can benefit from these resources.
Well, you fantastically summarized the spirit of HCA. We want to make these things open and share them across the world, even though there's industry bias, or there's maybe GDPR, all these constraints. We thank you very much for that discussion. I think that was really fun, and yes,
thank you very much.